Okay, so hello students, welcome to the sexual reproduction in flowering plants, lecture number 6. So today the topics that we are going to cover include a recap. We'll do an initial recap of some of the things that we have learned in lecture 5. Actually, I did a bit of more research and I wanted to share some more content or some more topics, some more information, some more analysis on the some of the points on lecture 5. It's not going to take much of time. The next we'll proceed on to fertilization, the double fertilization. Then we'll move on to the post-fertilization changes. We'll study about endosperm and study about embryo as much as possible. And the next lecture, that is lecture 7, is going to be the last lecture for this chapter. I will complete with the, if any part of embryo is remaining, we'll cover that. And then we will study seed and complete with apomixis and polyembryonic. So, okay, so just let's start. First, I would just like to give a brief recap. So exactly. So where are we positioned now? Okay. So we have the male reproductive hole, the androecium, the female reproductive hole, the gynoecium. So now both of them have matured. They have formed like both of like the anther, the pollen grain has matured. The stigma surface is receptive now. So both of them. Just a second. So both of them have attained maturity. Okay. So now the next step is pollination. We have studied the pollination, the pollinating agents, everything in detail. So then the next step was self-incompatibility. You must remember. And there's another thing which is prepotency. So I had mentioned the whole phenomenon of prepotency, but I hadn't mentioned the exact term. So I would just like to mention the term here. So prepotency is like promoting pollen germination of certain compatible pollens. So we had discussed this in complete detail that the pollens, they're falling on the stigmatic surface. So now they can be compatible or non-compatible. So if they're incompatible, whatever we say, if they're incompatible pollens, then what we will observe self-incompatibility. That is, the stigmatic surface is retarding the pollen germination. But in case the pollen that has fallen on the stigmatic surface is compatible, we will observe that the pollen germination is being promoted. So I just added a name for this, which is prepotency. Okay. So next was pollen tube growth. So yeah, so I would just like to add some points to this. So we had learned. So let me draw this. So see, this is the stigmatic surface, and these are the cells present over here. Okay, these cells are known as a transmission tissue. On top of this, the pollen grain has fallen like this. Another pollen germination is happening. So now the pollen germination is happening through the pollen, through the stigma, then through style, through the transmission tissue. So the new term here is the transmission tissue. And the next thing is, okay, so the transmission tissue is, if it's soft parenchymatous tissue. Okay. And what is it? So, okay, let me just write it over here. Okay, so this part is clear, right? The pole. So from this pollen, the pollen tube that is emerging, it is going downwards. Okay. So the tip of the pollen secretes pectinase and cellulase to destroy the transmission tissue. And the next is the structure of the growing tip of the pollen tube. It's something like this. So I'm just drawing the diagram and then I'll explain. Callose plugging. This is EC cell wall. So the next thing is, so now the pollen, so after this pollen pistil interaction thing, what are we going to observe? We are going to observe the growth of pollen tube to the stigma style, and then it goes to ovary and ovule. So now what are we, so just what I'm adding that the stigma and style are composed of transmission tissue, which is essentially soft parenchymatous tissue. Now, the tip of the growing pollen tube, it secretes pectinase and cellulase, which destroys this transmission tissue. Now, the thing is that it is it is also going to destroy the tip of the growing pollen tube, right? Because it is like if you just go to the composition of the same, so it is like it is also supposed to be destroyed, right? Because if it is secreting pectinase and cellulase, it is going to destroy the pectin and cellulose composition. So what is the remedy that we have? We observe that in the growing pollen tube. So this is a growing pollen tube. We are observing that there is the pectocellulosic wall. That's very fine. We have the pectocellulosic wall on both sides. Okay, so the diagram is getting worse. So just observe that we have the growing pollen tube. We have the pectocellulosic wall. So PC hall means PC is essentially pectocellulosic. Okay, it's right. Pectocellulosic. We have the pectocellulosic wall throughout the length of the pollen tube, except at the tip of the pollen tube. Except at the tip. See, the tip is devoid of the pectocellulosic content. So that's why the tip of the pollen is secreting pectinase and cellulase. It is destroying the transmission tissue, but it is not causing self-destruction, right? So the reason being pectocellulosic wall is absent at the tip of the pollen. So the next is the male gametes I had mentioned during the growth of the pollen tube through the style. The male gametes are formed. The generative cell, they divide to form the male gametes. And what we observe is callose plugging. What is callose plugging? So so essentially what are we observing? That the pollen tube is growing downwards. So by any chance, the male gametes should not backflow through the pollen tube. So to prevent the backflow, there is callose plugging. To prevent backflow of male gametes. I think this is fairly understood, right? To prevent the backflow of male gametes, we have the callose plugging. Okay. So the pollen tube is growing like this. The male gametes should not go up like backflow. So to prevent, we have the callose plugging. Okay. And the next, after the growth of the pollen tube, the stigma and style, it has to enter the ovule. We had discussed that porogamy, chalazogamy, and mesogamy. We had discussed. And just and what I'm adding is adding the examples. Okay. So porogamy, most common in angiosperm. What does it do? Enter the ovule through micropyle. The next is, okay, so let me give a bullet point over here. Chalazogamy. Example is casuarina. The pollen tubes enter through the chalaza. I'm just writing chalaza. So, and the last thing is mesogamy, which is observed in cucurbita. Cucurbita, okay, whatever, cucurbita. And it enters through the integument. Okay, so I had discussed this in detail. I just added examples, okay, which probably I had missed in the previous lecture. So what happens in porogamy? The growing pollen tube enters the ovule through the micropyle. This is the most common in angiosperms. In chalazogamy, the pollen tube enters the ovule through the chalaza, okay, and in mesogamy, it enters the ovule through the integuments. But we have to keep this in mind that entry to embryo sac is always via micropyle. This is the highlight point that I had repeatedly mentioned in the previous lecture, that even though we are observing the variation in case when the pollen tube is entering the ovule, it might be through micropyle or chalaza or integument. But if we talk of embryo sac, it always has to enter through the micropyle because it is because the embryo sac is covered by integuments on all its sides, so you can't break through it. Okay, So to get into the ovule, you have to go through the macropyle route. Done. So now we have completed the first part. So recap, let's just mark it in red, is done. What did I do? Exactly, I introduced the term prepotency. I described the phenomenon. I just mentioned the term over here. I introduced the concept of transmission tissue, and I did this callus pegging thing. I discussed the growing of pollen tube through the style in more detail. And in the end, I just added a few examples for the different for porogamy, chalazogamy, and mesogamy. So now we are good to go. We will move to the second part of what we were supposed to discuss, that is the double fertilization. Okay, so before getting into double fertilization, let's just learn about pre-fertilization events. No, I know pre-fertilization event was included everything that we have studied till now. I'm talking of the immediate events, like just the events that are just preceding the event of fertilization. Okay, events occurring just before fertilization. So the first point is one synergid completely degenerates. Okay, so we had two synergids. You have to remember that the synergid had filiform apparatus which guided the pollen tube into the embryo sac. Okay, but you have to remember that just before fertilization, one of the synergid gets completely destroyed to provide space for the entry of the gametes. Okay, so complete degenerates to provide space for gamete entry. Okay, so that the gamete can enter the embryo sac, one of the synergid is completely degenerated. So the next step is, okay, we have, so you have to remember the structure of embryo sac. We had a 7-celled 8-nucleate structure. So in the central cell, you observed that there were 2 polar nuclei. Okay, they get fused to form secondary nucleus, also known as a definitive nucleus. Okay, so now the pollen tube. So the pollen tube has gone through the whole stigma style. It has reached the embryo sac. So the pollen tube, you know, it merges with, like, it attaches with the embryo sac. Now the male gamete has to go in. So there is both the synergids. They have the filiform apparatus which guides the pollen tube, and one of the synergid gets completely degenerated to provide entry space for the gamete. Next, what happens in the central cell? You have 2 polar nuclei, that is, let me write it as N plus N. They get fused together to form the secondary nucleus 2N. This is also known as a definitive nucleus. Okay, so now this is the backdrop. Now we are getting to the actual event. And so now when we will discuss the actual event of photosynthesis, you will more clearly understand why I am referring to this process double fertilization. So now see, this is the like the fun thing. So we had 2 male gametes. So one, okay, let me just continue in red. So there is one male gamete. It is going and fusing with the egg or ovum, whatever you say. Okay, and it's forming the zygote. That is 2N. See, it is also important to note the ploidy level of each of the participating characters. Okay, let me just tell it character. Okay, so just take note of the ploidy level of each of the participating members of this whole double fertilization process. So you have one male gamete, N. Okay, it is going and fertilizing with the ovum, that is also N, resulting in the formation of zygote. Okay, this is the normal fertilization, right? This is normal fertilization, which we refer to as syngamy. Okay, this is something I think we all know, right? We all are familiar with this, that male gamete and female gamete, there is egg, ovum, or sperm. Sperm is a male gamete. Right? Sperm is a male gamete, the female gamete, the egg and ovum. They fuse together, the fertilization occurs, resulting in the formation of zygote. This is syngamy. And in case of plants, what are we observing? We have one male gamete and we have the ovum. They're fusing together, they're forming zygote. This process is known as syngamy. Now here is the another special thing which is known as triple fusion. Here what happens, I guess you might have already guessed it. So we have this another male gamete which fertilizes, which fuses with the definitive nucleus, or you can say secondary nucleus, the same thing, resulting to the formation of, okay, just draw the same kind of arrow then, triploid primary endosperm nucleus. That is 3N. So see what is exactly happening. So you remember we had 3 antipodal cells on the top, each containing one nucleus. Here we had the central cell. We had 2 nucleus in that. Here we had 2 synergids and an ovum. So when the pollen tube is getting in, one of the synergids is getting degenerated. So now we have 2 male gametes. One of the male gamete goes and fertilizes with the ovum. Well and good. Forming zygote. This process is known as syngamy. Okay. And the other male gamete, it goes and fertilizes with the definitive nucleus. The 2 polar nuclei in the central cell, they had fused to form the secondary nucleus or the definitive nucleus 2N. So the another male gamete N goes and fertilizes or combines with this definitive nucleus 2N, leading to the formation of triploid 3N primary endosperm nucleus. This process is known as triple fusion. Okay. So syngamy plus triple fusion is equal to double fertilization. Okay, so now we are done with the double fertilization. So see why it is known as double fertilization? Because we are observing 2 fertilization events operating simultaneously here. First is the syngamy, second is the triple fusion. In syngamy, what is happening? One of the male gametes, N, is combining, is fertilizing the egg ovum, that is also N, resulting in the formation of 2N zygote. In the case of triple fusion, what we are observing? We have another male gamete N, which is fusing, combining with the definitive nucleus. Definitive nucleus is 2N. So N plus 2N, the combined to form 3N, which is the triploid primary endosperm nucleus. Okay. Now in the post-fertilization events, we are going to study this, how the zygote develops into the embryo, and this primary endosperm nucleus forms the endosperm. This is the next topic of discussion, that is the post-fertilization events. So the syngamy and triple fusion, they are togetherly known as the double fertilization. Okay. So after fertilization, synergids and antipodal cells are destroyed. Okay. So one synergid had been destroyed when the pollen tube was getting into the embryo sac to provide space for the male gametes, and the another synergid and the remaining 3 antipodal cells, they are destroyed after double fertilization. Okay. So double fertilization also done and dusted. So give a clap for yourself. You have completed double fertilization as well. So we have done the next topic as well, which is double fertilization. Done. Okay. So just a quick recap. So what is happening in double fertilization? Syngamy and triple fusion. Syngamy, male gamete plus ovum, N plus N, forming zygote 2N. Triple fusion, another male gamete N plus definitive nucleus 2N. What is definitive nucleus? The 2 polar nuclei in the central cell, they were combining just before fertilization to form definitive nucleus or secondary nucleus 2N, N plus 2N, triploid 3N primary endosperm nucleus. Now the zygote will develop into the embryo. Primary endosperm nucleus will form the endosperm, which are we going to study in the post-fertilization events. So which is our next topic of discussion. Okay. So now the broad heading, post-fertilization events. So next we have the post-fertilization events, and the 2 main constituents of the post-fertilization events, they include, one is the endosperm development and embryo development. So let me just write it. So these are the two key events that are happening post double fertilization: endosperm development and embryo development. Okay, so see the sequence is also important here. You know, endosperm development precedes embryo development. So first endosperm has to be developed, then embryo will be developed. The only reason being the availability of nutrition. So endosperm provides the food, nourishment, nutrition to the developing embryo. So the endosperm has to be present so that the growth of the embryo is smooth and optimal. So that's why endosperm development precedes embryo development. So this is important. So now we are coming to endosperm development. So first, okay, so let me just rewrite this. So first point is, what is endosperm? The endosperm is a nutritive tissue. What is its function? Provide nourishment to the developing. So the so exactly the after triple fusion, we are observing triploid primary endosperm cell, primary endosperm nucleus, sorry, which would gradually after the post fertilization changes develop into the endosperm. It is a nutritive tissue, and its the primary function is to provide nourishment to the developing embryo. Okay, so I have mentioned this point. Let me just write it. Okay, so yeah, I discussed this. Endosperm development precedes embryo development. The only reason being, so the function of endosperm is to provide nourishment to embryo. So when the embryo is growing for its optimal growth, we require the presence of endosperm. So that's why endosperm development occurs before embryo development. So the next we are having is the types of endosperm based on development. So the first one is free nuclear endosperm. So you will find the mention of all these terminologies in NCERT, and I would discuss all of them. Okay. I am not much a big fan of writing definitions throughout. I was mentioning the primary important points. It's which would be sufficient for you guys to understand the concept. Okay. So what is happening in free nuclear division? Let me just draw the diagram. Okay. So now I think you guys will yourself be able to just name the processes. So first we have, this is the PEN, primary endosperm nucleus. This is the zygote. So in the next step, what is happening? We are observing free nuclear division. So the number of primary endosperm nucleus is increasing, but it's not followed by cytokinesis. Okay. So this is a zygote. This is so this one is a zygote, and this zygote denominated Z, and these one are the PENs. The next step is, see, vacuolation or vacuole formation. So now this is the vacuole, and the last step is cytokinesis. So this is the free nuclear endosperm. So I just understood the process. You have the initial embryo sac, you have the zygote, you have the PEN, primary endosperm nucleus. So first we are observing free nuclear division of the primary endosperm nucleus, then followed by vacuolation, then followed by cytokinesis, leading to the complete development of the free nuclear endosperm. So now examples. Example, it is found in case of cotton and maize. Okay, so done. The next is cellular endosperm. Okay, so I think this is easier. So you have the pen. This is the zygote. The initial structures are just the same. You have the pen, you have the zygote. Next, the first step you're observing, just take a note of this, is normal mitosis. Normal mitotic division, that's it. Next, it is followed by random mitosis. So see, mitosis means there is both karyokinesis and cytokinesis, and that's why we are observing this kind of structure. So I think you guys will personally, like by yourselves, you will be able to differentiate between the free nuclear endosperm and cellular endosperm. So let aside the way, the method of formation of either of them. Even by looking at the structure, you can say the difference. So what is absent here? Vacuole is absent here, right? Vacuole is absent here. So you have to take note of this. So in case of cellular endosperm, what is happening? We have the initial condition, we have a pen, we have a zygote. So the pen is first normal mitotically dividing and followed by random mitosis, resulting in the formation of this beautiful cellular endosperm. So what is this example? Found in case of petunia. Okay. So let me just add some, highlight points about this. Okay, no. Let's not go this way. Let's first complete the types of endosperm first, and then we will get on to some interesting informations about either of them. So the last is the halobial endosperm. So the first one was free nuclear endosperm. What did we observe? The pen was first undergoing free nuclear division, followed by vacuolation. Then it was followed by cytokinesis. Then the final whole structure was ready. In case of cellular endosperm, we observed that the PEN underwent a normal mitosis followed by random mitosis. Now in halobial endosperm, we are going to observe a combination of them both. So this is 2N, this is the pen. The first step is normal mitosis. Okay. Then it is followed by free nuclear division. So just observe all the structures that I'm drawing. Okay. It's more than sufficient. I believe that this is more than sufficient for you guys to understand as well as remember the phenomenon that is happening. So see, first we are observing normal mitosis, just like in case of cellular. Then we are observing free nuclear division. Then what follows free nuclear division is obviously you guys are already aware of that, vacuole formation. And the last step, obviously, you get it right, cell wall formation, ready leading to the formation of our halobial endosperm. So now our halobial endosperm is ready for action, if we can say so. So yeah, our body is now ready for action. Okay, so see, beautiful, isn't it? So we are observing the cellular part as well. We are having the free nuclear part as well. This is your halobial endosperm. Now is example found in halobialus. Okay. So what did we observe? So first we observe what is endosperm. Endosperm is essentially the nutritive tissue. Okay, it is the nutritive tissue which is present in the embryo sac. Okay, so its function, obviously it is nutritive tissue. Its function is to provide nourishment to the developing embryo. The second point you have to keep in mind is endosperm development occurs before embryo development, so that there is an abundant supply of nourishment and food reserves to the developing embryo for its optimal growth. So endosperm growth occurs before embryo development. So the next we classified embryo sac based on their method of formation. We observed free nuclear endosperm, where initially the PEN underwent free nuclear division, then followed by vacuolation and at last cell wall formation. Then we observed cellular endosperm, where we observed that the PN underwent normal mitosis, followed by random mitosis, leading to the formation of cellular endosperm. Then we observed the halobial endosperm, where initially we had normal mitosis, then we had free nuclear division, followed by vacuolation and cell wall formation. Okay, so now we have discussed and completed in detail the endosperm formation. Now some interesting facts about them. Let me just write facts, but write factors. Okay, you can call it my lingo, linguistics. Okay, factors. You have to keep these factors in mind. Okay, most common endosperm type. So if you guys want to make any guesses, you can just pause the video and just try and hit some random guesses. But the most common endosperm type, I'm just directly going to the answer. It's free nuclear endosperm. Free nuclear endosperm is the most common type of endosperm. And the next is coconut water. Free nuclear endosperm. And is and the okay, let me write it in this way. So I guess you all must have drank water from coconut. Okay. So what do we have? So the coconut water is essentially free nuclear endosperm, but the white kernel surrounding that present in the tender coconut is cellular endosperm. So see, just witness the beauty. So you initially you have free nuclear endosperm in the form of the coconut water, and you have cellular endosperm in the form of the white kernel surrounding it. So this is the beauty of nature. Okay, so you just keep this two facts in mind. They're present in NCERT as well. The most common endosperm type is free nuclear, and we have coconut water as free nuclear endosperm and white kernel in the tender coconut as cellular endosperm. Okay, so we are going in considerably good pace today, and we have completed our endosperm development as well. So again, give a clap for yourself, guys. You've completed endosperm as well. So now we are coming to the last portion, that is embryo. Post embryo, we are going to discuss about. Okay, I think there has been. I have to change the order of the pages, perhaps. So now the last part is embryogenesis, that is the development of embryo. For the development, we are going to study about seed development and food development. They are easy topics. Okay, they are easy topics, and I believe that everyone has a brief idea of this because we have studied in the 8th and 9th grade. So we have embryo development as today's last topic, and thereafter in the next lecture we will complete fruit, seed development, apomixis, polyembryonic, and complete this chapter. So now we have embryo development. So I'm just writing in points. The first point is embryo develops at the micropylar end of the embryo sac. I had told you guys to completely, perhaps make a chart if possible, that where it is the chalazion end involved and where it is the micropylar end. Like you have to remember, we had 4 megaspores out of the one functional megaspore, this and that. Then we had this embryo development, then entry of pollen tube. We have a lot of stuff. We have the orientation of the nucleus and vacuole in ovum and synagid, etc., etc. You have a lot of stuff which is essentially based on the direction, based on the micropylar end or chalazion end. So you can make a table of your own and just note down this happens. This particular structure is present towards the micropylar end. This particular event happens at the micropylar end. Okay, so the embryo development occurs at the micropylar end. Okay. I had already mentioned the fact that endosperm formation occurs before embryogenesis. So now, after a bit of endosperm is formed, then only the zygote starts forming the embryo very clearly. Unless you don't have the resources to provide to the developing embryo, there's no point in starting the division. So first we have endosperm development, then we have the embryo development. So what exactly happens? The zygote gives rise to the proembryo and subsequently globular heart-shaped mature embryo. So like this is very elementary stuff. The zygote develops into the embryo, the PEN develops into the endosperm. Okay, so the zygote gives rise to proembryo, and this proembryo develops into embryo, and we have some adjectives given for the embryo. It is globular, heart-shaped, and mature. So now let's get to the mechanism. Okay, mechanism is a bit long. Okay, and perhaps it will go over more than 2 pages. Okay, so the first is we have the zygote. Then we are having the first division is a transverse division. Transverse division is TD. Oh, it is TD. TD. Okay. Aha. Just take a note. This is unequal transverse division. So now what does it form? It forms this. Now we have a basal cell. We have a terminal cell. Okay. So you have the zygote. It undergoes unequal transverse division, leading to the formation of basal cell and terminal cell. Then we have, now you guys will yourselves be able to tell what exactly is happening. See, the basal cell, let me write it in red. The basal cell undergoes TD, that is transverse division. And the terminal cell TC is undergoing LD, that is longitudinal division. See, so the terminal cell this is undergoing this kind of division, leading to the formation of this. And the basal cell is undergoing further TD transverse division, leading to the formation of this. Okay, this is known as the basal dyad because obviously two cells are present, and this is the terminal dyad. Okay, so let me just so TD I have mentioned transverse division, and LD is longitudinal. Okay, okay. Just do another exercise. Known. See, just take count of this. So this is the first division. This is the second division. Now we are moving on to the third division. What exactly is happening? So now we have the basal cell undergoing transverse division, and the T cell, terminal cell, undergoing another longitudinal division. So now what the structure is gonna be is gonna be something like this. So see, this part is easy, I guess. So you had the initial zygote. It underwent unequal transverse division, leading to the formation of a basal cell and a terminal cell. Now the basal cell is continuously undergoing transverse division for the next 2 steps, but the terminal cell is undergoing longitudinal division. So okay, so this is number 3 division, right? So the first division was a transverse division. The zygote formed, the basal cell and the terminal cell. So for the next 2 steps, second division and third division, the basal cell underwent transverse division and led to the formation of basal tetrad, like basal cell, basal dyad, basal tetrad. Dyad, tetrad, you can understand why we are referring them as. So considering the number of cells, and the terminal cell is undergoing longitudinal division, LD, in the consequent second and third division, leading to the formation of terminal tetrad. Okay. Now, same retelecast. Let me just write it. A retelecast for division number 4. So what is essentially the basal? Basal cell, they're undergoing transverse division, and the terminal cell, they're undergoing longitudinal division. I think you can understand what these short forms are referring to. BC basal cell, TC terminal cell, TD transverse division, LD longitudinal division. So now we are having this kind of structure. So now earlier we had tetrad, now you will have an octant. So the TC terminal cell is again going longitudinal division. So you will observe this kind of a structure, and the basal tetrad will now form another basal octet, and it is again going transverse division. So you are going to observe 8 cells. So 1, 2, 3, 4, 5, 6, 7, 1, 2, 3, 4, 5, 6, 7, 8. So 1, 2, 3, 4, 5, 6, 7, 8. Okay. So now we are having an interesting observation. Okay. So now, okay. So I think it's very clear up to here. So we had exactly 4 stages. The first, this is the initial zygote. Let me just write it. There's the initial zygote in bracket initial condition. We had the initial zygote. It underwent unequal transverse division forming basal cell and terminal cell. The basal in the consequent next 3 divisions, in fact, the basal cell continuously underwent transverse divisions forming basal dyad, basal tetrad, and basal octant, and the terminal cell underwent consequent longitudinal divisions forming the terminal dyad, terminal tetrad, terminal octet, and we finally observed and we finally came up to this structure. Now it's time for the 5th division, which is the most interesting of all. Okay, let's just take a new page for this. So what is happening? Division number 5. What is happening? Only terminal basal cells are undergoing transverse division and the T cells are undergoing periclinal division. Okay, so we had basal octant. Of them, only the terminal one. So what am I referring to the terminal ones? Let me see. This is the terminal one. This is the terminal one. So only 2 entities, they are further undergoing transverse division. Okay. So now let me just draw the final structure for you guys. 2, 3, 4, 5, 6, 7, 8, 9, 10. You had 8, only 2 of them are dividing, so just add more to 2, 3, 4, 5, 6, 7, 8, 9, 10. And now it has undergone periclinal division. So maybe you are having some difficulty understanding what is it. Once I draw the diagram, it's going to be clear for you guys what exactly is being referred to here. Okay, and just so this is like the dotted is exactly at the back. So this is like something like this. You're watching it from here. So you're observing the front view, and this is hidden view. Okay. So this is the final structure that we're ending at. So first zygote, initial zygote underwent unequal transverse division forming one basal cell and a terminal cell. The basal cell and terminal cell respectively underwent 3 consecutive, where the basal cell underwent transverse division and the terminal cell underwent longitudinal division, resulting in the formation of basal octant and terminal octant. And then in the division number 5, we observe that only the terminal basal cells, they underwent transverse division. So now we have 10 basal cells. This is the unit. And in the transverse cell octant, we observe periclinal division, leading to the formation of this structure. Now it's time for naming each of them. Postoria or vesicular cell. Okay, this cell is known as hypophysis. Okay, hypophysis is the joining link between okay. Okay, let me just mention this over here. So I was thinking where I would mention this. This is the epibasal layer. This is the epibasal layer, and the inner one, obviously, I think you can guess it already, is the hypobasal layer. So we have see the topmost cell. I have drawn it a little bit bigger. This is the haustoria, the vesicular cell. Which are the hypophysis, the connector between this suspensor and the 16-cell globular membrane. I'm writing it. Okay. We have the epibasal layer and the hypobasal layer. Now, what I was referring to, this is essentially our suspensor. This is a suspensor, and this is exactly the 16 celled globular embryo. Okay, we have now reached the stage where the zygote has undergone various kinds of division. We observe transverse division, longitudinal division, periclinal division in different steps. Okay, 5 steps in total. And now we have a suspensor consisting of 10 basal cells and have a 16-celled globular embryo. This is where we are ending. And so now what will happen? This thing will only go on, will proceed further, leading to the formation of cotyledon. Okay, this is where we end this lecture. In the next lecture, we are gonna cover the functions of suspensor. We are gonna cover about the dicotyledonous and monocotyledonous embryo. This is a very short topics. It won't take much of a time. The important part of embryo was the formation of embryo. This formation of this suspensor and 16-cell globular embryo structure. We have completed the formation part. Okay, and the next we'll get on to seed, fruit development, and at the end apomixis and polyembryony. And so in the next lecture, we are going to complete this chapter. I hope you guys had a good time learning, a good learning experience. Okay. After completion of this exercise, after viewing this lecture, do read the NCERT once. Okay. I think it's gonna be, it is gonna be helpful. So we have also done. So we have completed whatever topics we had planned to do today. Okay. So yeah. So how we observed. So I'm just giving you a quick recap. We initially learned about prepotency, how then the growth of pollen tube through the style. Okay, in the next slide, in the next case, we just I gave you the examples of orogamy, chalazogamy, and mesogamy, and then we got onto double fertilization, the events just prior to fertilization. Then we got onto double fertilization. Double fertilization is syngamy plus triple fusion. Okay, we have studied that in detail. The next we moved on to the post-fertilization events, which include endosperm development and embryo development. And endosperm development precedes embryo development for sure. We studied what is embryo, what is its function. We studied the types of embryo based on development, pre-nuclear endosperm, cellular endosperm, halobial endosperm, and we studied each of the mechanisms of formation of them in detail. And the last, we moved on to embryo development, where we observed how from a single zygote via five sequences of division, we obtained a suspensor and 16-cell globular embryo structure. So this is where we end today's lecture. Thank you guys.